Okay, we're going to have a look at glucagon now and the effect it has on the target cell. So the target cells are usually the liver cells and also skeletal muscle cells. Let's think about liver cells, and we know that when glucagon binds to the surface of a liver cell, it's a peptide hormone, so we can't get inside it. It's going to increase glycogenolysis, break down the glucose from glucose 1-phosphate, and it will also increase gluconeogenesis. So that's making glucose from new molecules of glucose from other precursors. Now, we're not going to talk about gluconeogenesis in this video. We're going to concentrate on how receptor binding leads to glycogenolysis, and we'll leave the gluconeogenesis to a later one. All right, so we've got a problem is with polypeptide hormones is that you've got a cell there and you have a receptor on the surface. We know that the glucagon molecule is going to come and bind to that receptor, and then that somehow is going to cause changes inside the cell, which leads to the hydrolysis of glycogen. How is that receptor binding transmitted to the inside of the cell? Right, there is something called a second messenger, and that second messenger, we'll see what it is, but it is a compound called cyclic AMP. So you've heard of ATP, adenosine triphosphate. This is adenosine monophosphate. Show the structure of it in a bit, but that is a second messenger. So that the concentration of that increases inside the cell, and then this stuff has works its magic and causes the increase in glycogenolysis and also gluconeogenesis, but we're just going to talk about glycogenolysis in this video. Okay, so I should get rid of that. All right, so what I've drawn here is a bit of a cell membrane, and we have got, this is supposed to be a glucagon receptor. I've also drawn this green one, which is supposed to be adrenaline receptor, because they work in exactly the same way. Adrenaline binding to a liver cell will also increase glycogenolysis by the same pathway, by this cyclic AMP and so forth. And this other red protein I've drawn here is the enzyme which makes cyclic AMP, and that's called adenylate cyclase. So the sequence of events is you get glucagon coming along and binding to the glucagon receptor, or in fact, it might be adrenaline binding to the adrenaline receptor. That causes certain changes to the glucagon receptor, changes shape, and then it interacts with the enzyme adenylate cyclase. Adenylate cyclase then makes AMP. So we just have a quick look at how that works. Right, so hopefully you will recognize this structure here on the left as being ATP. So you've got, your adenine, yeah, you've got ribose sugar, and then you've got 3 phosphates. Okay, so what adenylate cyclase does is it will convert ATP into cyclic AMP, which I've drawn here. And you can see that the phosphate group is, on ATP, it's stuck to carbon number 5. One of these, that goes around and joins onto carbon number 3 there, and that's cyclic AMP. Right. And once you've made cyclic AMP by adenylate cyclase inside the cell, that then, there's a whole cascade of things which happen. So we'll have a look at those, okay? Now, this is pretty complicated, and there's quite a lot of enzymes here, and they've all got very similar names, which makes it even more confusing. So let's go through it slowly. Right, there's this enzyme here called Protein Kinase A, and that was actually discovered by Krebs, as in the Krebs cycle, and he got the Nobel Prize for discovering how that works. Okay, it's an enzyme. It's inactive unless you get cyclic AMP binding to it. Okay, cyclic AMP binds to it, it becomes active, and what it does is protein kinase A, what it will do is it will stick phosphate groups onto proteins. And the way it gets the phosphate from an ATP molecule and transfers it from an ATP onto the protein. You make ADP in the process, see there. Now, one of the proteins which it targets is this enzyme, which I've drawn in blue here, called phosphorylase kinase. That's another kinase enzyme. Kinase is the name for an enzyme that sticks phosphates onto other things from ATP. So phosphorylase kinase that's inactive there. But once pKa has stuck this phosphate onto it, it becomes active. That can then act on its own substrates. And the substrate that this phosphorylase kinase will act upon, shown with this green arrow here, right? The substrate it acts upon is glycogen phosphorylase. Now, glycogen phosphorylase is the enzyme that snips a glucose 1-phosphate off the end of the glycogen polymer. So you've got glycogen phosphorylase B, that's inactive, that can't do anything. But phosphorylase kinase transfers, again, there's a bit of a pattern going on here, transfers this phosphate group onto it, and that activates glycogen phosphorylase. It turns into the A form, which is then active. And what glycogen phosphorylase does is this. So I've drawn here, this is a glycogen molecule. Glycogen in the liver cell, stored in the liver cell. I've only drawn it with 4 glucose molecules long. It's many hundreds, really. And they're joined together by these glycosidic 1,4 linkages there. Right, so what happens is that an inorganic phosphate, so that's PO4, you can usually draw it just with a little circle around that, but really it's PO4 3-. What that does is that actually attacks this oxygen here, sticks a phosphate group onto that, as you can see there, to form glucose 1-phosphate. Glucose 1-phosphate. And the glycogen molecule here is now 1 shorter, so I'll call that glycogen N-1. Okay, and we form the glucose 1-phosphate. There's another enzyme then which can hydrolyze off the phosphate group, and you get glucose, which is then released into the bloodstream and can be used by other tissues in the body. In the case of skeletal muscle, this glucose 1-phosphate, you don't lose the phosphate off the end there. That will turn into glucose 6-phosphate, and it will go into glycolysis. So the way it's called a signal transduction pathway, the signal being the binding of glucose onto the receptor, and then the final thing is for the glycogen phosphorylase enzyme to be active. But of course there are lots of other signals, intermediates in the way, the cyclic AMP, which then activates protein kinase A, and so on. Right, in the next video we'll talk about how this signal transduction pathway can lead to gluconeogenesis. That's a bit more complicated than that, make it a bit of a too long a video to do it all in one go, so we'll do that in the next one.